Okay, perfect. So we will start with what is first. So we'll go describe the molecular characteristics of the contractile filaments found within the functional unit of the skeletal muscle cell, the sarcomere. Okay, so we'll start with that right there. So we'll go up to some pictures real quick. Okay, so in a muscle cell, we have, various units called sarcomeres. And a sarcomere is basically here's a good example. So here's one of my drawings. So between Z discs, so this is a Z disc. I believe it's just a protein structure, I think. Anyway, this is another Z disc, this is another Z disc. So in between Z discs are sarcomeres. And within sarcomeres, we find these different structures and different filaments. So we have this big important one called myosin that plays a critical role in muscle contraction, as well as actin. So we'll dive deep into each of the structures a little more. So with myosin is made up of two heavy chains, which makes up the tails and the head. And then there's these light chains as well. And we won't talk about the light chains until later. But, and again, this is skeletal muscle. We're talking about skeletal muscle first. So that's myosin that plays an important role, as well as this actin, both types of filaments. So actin is made up of G-actin, which is just individual kind of units. And when they're put together in a row, that's called F-actin. So it creates this kind of helical structure, like what I showed above, like right here. Anyway, so those are the big players in muscle contraction of skeletal muscle. In addition, we have what's called troponin that also plays a very important role, and tropomyosin. So troponin is made up of three subunits, the C, T, and I subunit, and we'll explain what each of those does here in a second when we go through the process. Tropomyosin is what wraps around that helical structure of actin and blocks it from these heads attaching. Anyways, that's a little, I guess these aren't as important, but titin is what anchors myosin to the walls, like what we see up here. And then nebulin, I believe, is just where the actin attaches. Anyway, so now going into the mechanism of it. So basically what happens, calcium comes in and an action potential is generated. So again, I don't think we need to go too much into synapses, and we talked about that last time in the last video. But action potential comes in, calcium rushes in, binds to the snare proteins. Snare proteins release neurotransmitters. Neurotransmitters come and bind to nicotinic 1 receptors, allowing a continuation of that action potential. And then the action potential goes along the outside of the skeletal muscle, and on the skeletal muscle, there's these little indentations called T-tubules, and that's where we get a lot of our calcium. So we'll zoom in a little bit to one of those T-tubules. So action potential comes in, right here, and then it opens a bunch of voltage-gated channels. Main one we'll pay attention to is this one down here. So it opens this DHP receptor right here, and it's kind of, when this opens, it kind of forces this one open as well. Anyway, so it forces this one open, and this one is attached to the sarcoplasmic reticulum, which then allows calcium to rush in through that channel, that mechanically gated channel. And then the affinity for calcium down here inside of this cell is so high that it won't really go out here much. It'll just go straight down into the cell. So this is the inside down here. That's the inside of a skeletal muscle cell. So calcium rushes in, and it's able to go do its function. So what happens is that calcium comes in, and we have troponin, which has those 3 different subunits. So calcium comes in and binds to the C subunit, and then this allows the other subunit. So you got the C subunit, I and T. So the T attaches to tropomyosin and kind of pushes it out of the way. So the C one attaches to tropomyosin, pushes that away, and then the I one attaches to the actin. Anyway, so when it pushes the tropomyosin out of the way, it allows for these heads to come and bind. And so when these heads bind, then we get a process which is called the cross-bridge cycle. So what happens in the cross-bridge cycle is that calcium comes in, binds, we get that tropomyosin out of the way, and then we're able to bind. That head is able to bind. So with this ADP and this phosphate, it's able to bind. And to clarify, this is called a cocked position, and this down here is called an uncocked. So we'll be using some of those terms. Anyway, calcium comes in, it's able to bind now finally. And then when it binds, when those two things get pulled together, that whole process of binding and then unbinding, or binding, becoming cocked. So it's kind of like it's pushing the actin and the myosin, or it's pushing the actin together. Sorry, it's hard to explain without like using my hands to show you. But so it basically pushes these together. So these black lines that we see here kind of pushes them all together, pushes all this together, and that is what contracting your muscle is. So again, it's all getting it's shortening, and it's these heads that bind on and then push them together, which anyway, harder to explain without my hands or without motion, but that's the basic idea. Anyway, back to the cross-bridge cycle. So it binds, and then we lose ADP and this phosphate first, and then ADP, and then energy and heat is released. Then we just have this head, this empty head, this bare head in the uncocked position that is binds. But then we need this thing to release. So we add ATP. ATP, when this binds, ATP is always what causes this to become cocked again. So to go back to how it was. And then to start the whole process over, we need an ATP to turn into an ADP and a phosphate. So that's where hydrolysis comes in. So pretty simple cross-bridge cycle. Okay, let's go to the next question, make sure I got all that. So, okay, we did that. The mechanism, went through that. Okay, so energy sources for muscle contraction. So energy sources come from a few different places. One, so we have aerobic and anaerobic places where they come from. So anaerobic places, which means it doesn't use oxygen, is like the cytoplasm. So we got cytoplasm stores of, again, where ATP, that's the ATP energy we need. So ATP can come from cytoplasm, it can come from creatine phosphate, or glycolysis, if we're talking about anaerobic stuff. The cytoplasm, if it comes there, it can only last for 1 to 5 seconds. This one lasts for 10 seconds. This one lasts for 60. But then the best and most effective is aerobic respiration. So it uses oxygen coming from the mitochondria, and this can last for hours. And so that'll make more sense when we talk about the different types of muscle fibers and where they get their energy sources. I believe that's the next question, so we'll go ahead and go into that. So these muscle fiber types, I think it just asks to explain the main 2 types. So these aren't this isn't a main one. This middle one, we won't talk about that because only 5% is found in humans. Although it is kind of the most effective one. But we won't talk about that. So we'll just talk about these two types. So type 1 is known, so it's just type 1 muscle fiber type. And this one is slower. So if we think of like maybe cardio as opposed to, big explosive movements like jumping or whatnot. So cardio and let's say this. Let's use the example of pull-ups versus just hanging on a bar. So hanging on a bar would activate or would use these, which is a slow. And so basically what happens, I guess a few characteristics of these, its energy source comes from aerobic stuff, so it's able to last longer. So that means it doesn't fatigue as quick. So since it lasts longer, it doesn't fatigue as quick. So that's why like hanging on a bar, for example, you'll probably be able to last longer than doing pull-ups. So resistant to fatigue because it uses oxygen. And again, oxygen when the ATP source comes from aerobic respiration, then it lasts much longer, like we saw above, those different energy types or the energy sources. But again, not very explosive, so the energy out or the power output is pretty low. These are very red because the capillary density is high, because we need so much oxygen to go to those, so we need lots of blood flow to those areas. Myoglobin, I believe, is just it's associated with capillary density. I think it attributes to the color of the cells as well. Again, so very red cells here. Here for fast cells, so this is like explosive things like weightlifting and pull-ups and whatnot. So these use anaerobic respiration and again a big power output because we need them like weightlifting and pull-ups and things like that. However, since it comes from anaerobic, it fatigues pretty fast and it's very fatigable because again, those three energy sources that are anaerobic, don't last very long. We have the 5 seconds, we had the 1 to 5 seconds, 10 seconds, and 60 seconds. So it doesn't last very long. And since we don't need as much blood flow there, we have low levels of myoglobin and low capillary density. So those are probably the main things I would say that are important about those two different muscle fiber types. This third one, again, I don't think I need to talk about it, but it's basically a little bit of a mix. So it's fairly resistant to fatigue. It's got a medium output. It uses both, which is very useful. And so let me go mark some things off, make sure we're getting everything. The various sources of energy, relative amount of each source in the cell. Let's see. Advantages and disadvantages of each type under what condition might be. Okay. So yeah, we talked about the different conditions. So again here, maybe with more explosive things, more explosive types of exercise and whatnot, and here more like cardio or hanging on a bar or whatnot. Anyway, so I know this definitely depends a lot. It depends on genetics and gender, these fiber types, and where that energy is going to come from. So for example, women have more of that and men have more of that. That's why men are a little more explosive and whatnot than women are. Okay. Okay, so and those were let's see. 2 main types of smooth muscle. Describe a point of calcium entry. Okay, so now we'll move into smooth muscle. Okay, we'll go into smooth muscle now. So when talking about smooth muscle, we're going to compare and contrast this cross-bridge cycle. So as we saw in this cross-bridge cycle first, so we're going to be using that as kind of like a base point to explain. So this is skeletal, and these are smooth muscle. So we'll go into these different kinds. So we got phasic and tonic. So they're kind of different cycles we have that deal with the two different kinds of smooth muscle. So we'll start with the phasic. So again, we'll use the cross-bridge cycle as a template. So we have this head with the ADP and a phosphate. Calcium comes in. However, in this state, or in this step, we have MLCK that comes in, and this is a kinase. So this is where the light chains come in as well. So we mentioned how the myosin is made up of light chains and heavy chains, but how in skeletal we were mainly using heavy chains. Well, in this case, we're going to be using some of those light chains. So this is a light chain kinase, and kinases always add phosphates. So, and then we need an ATP to get that phosphate from. So, comes in, adds a phosphate. This extra phosphate makes this myosin head have a high affinity for actin, and so it's going to easily bind, and it doesn't really... Well, yeah, this alone is just going to push tropomyosin out of the way. So it's going to push tropomyosin out of the way, and that's what that extra phosphate does. And then, like we had in the step before, phosphate and ADP come off, which allows this to bind, but again, that phosphate is still hanging out there. And then once we're ready to have that unbound to the actin, we add ATP, which comes in and makes this get unbound from the actin. But again, that phosphate is still hanging on there. So basically what that phosphate does, another function, is that we can just kind of skip this finishing step. So when MLCP, which is a phosphatase, which takes off a phosphate, we only use this when we're done with the whole cycle. But the advantage of having this extra phosphate on, another advantage, is that we can just skip that step and come straight back over here and then just add the extra phosphate on. So we don't have to go through this whole step. Yeah, so this will just turn into, this right here will turn into an ADP and a phosphate, and we don't have to use as much ATP because that phosphate is still on there. So makes things pretty effective and not as fatigable, since we're not using as much ATP. So comparing that with this state, so this is the tonic phase, or the tonic cycle. And in the tonic cycle, we have the same steps. So MLCK and ATP come in. They add the extra phosphate. We see the extra phosphate here. However, in this case, MLCP comes in quicker. So basically, that phosphate comes on. It's able to move that tropomyosin out of the way. Or, excuse me. Yeah, pushes the troponin out of the way and allows this head to combine. Anyway, so after phosphate and ADP get kicked off, it'll bind. The removal of those two things make it bind. But then this MLCP takes that phosphate off, and what that does is it basically paves the way to this latch state. And this is really important because this latch state is basically when we have ATP come in, the lack of that phosphate allows the ATP to have a little bit more trouble binding, because if we remember, the binding of ATP is what makes this go back to the cocked position, which makes it come off. So if we need something that we need to stay contracted, if we need that smooth muscle contracted more, then we don't need that ATP on there just yet. And so the latch state is just kind of that phase in which it's hard for that ATP to bind. And so it's going to allow for longer contractions. So that's what the purpose of that whole thing is and what the latch state is. Okay, let me go back down and make sure I got this. Describe the two main types of smooth muscle. Okay, so calcium came in. Then when contraction ends, compare and contrast smooth and skeletal muscle, structurally, functionally, and regulation. So, again, it was highlighted a little bit in these stages, but the main difference is that skeletal muscle is what we can control, even though most of skeletal muscle is voluntary, or involuntary, which we'll go into. But skeletal muscle is some things we can control. Smooth muscle are things we cannot control. And with like things like a heartbeat, or well, I guess that's cardiac. But like blood vessels and various things like that are where smooth muscle comes in. We need a much greater energy. We need more energy for those because we need those to last longer. Blood vessels, if those got fatigued, and that might not be very good. So we need, or like our lungs, for example. So we need that smooth muscle to perform for longer amounts of time, which is why we have these stages right here that allow for a little bit more of the saving of energy. Again, with that extra phosphate on there and the latch state. So that's another difference between smooth and skeletal muscle. And yeah, so those are the main differences between skeletal muscle. Okay. So then we'll describe and explain muscle sensory receptors, muscle spindles, and the Golgi tendon. Okay. So with the stretch reflex, we'll come down here. So with stretch reflex, when we're talking about reflexes, it's skeletal muscle. However, reflexes are kind of involuntary. So again, what I said earlier, how muscles are, or skeletal muscle is oftentimes involuntary as well. So to explain that, we'll look at a muscle here. So if we have this muscle right here, inside each muscle, we have these things called muscle spindle apparatus, I don't know, a muscle spindle apparatus, but we have a bunch of them. And basically we have two motor neurons coming to this muscle, one to the muscle spindle apparatus and one to the alpha motor, one to just the muscle in general. This is the gamma motor neuron, this is the alpha motor neuron. And we have sensory going back. So what happens is that these two sensory neurons can detect stretching. So if we zoom in here on these sensory neurons, so we have these chain fibers and back fibers. The chain fiber is what attaches to the type 2A sensory neurons, and the back fibers is what attaches to the type 1A sensory. Basically, type 1A is activated when the muscle is stretching. So if you stretch your arm out, while your arm is stretching out, that 1A is being activated. And then once you're fully stretched, that 2A is activated. So that's when the muscle is stretched but the bone's not moving. Anyway, and so what that does, we'll scroll down here. So it will send that signal to the spinal cord. Okay, so if we have that sensory coming in, it will come to the spinal cord, and depending on the muscle that's being stretched, we will have different responses. So let's say your quad, for example, like in this picture, your quad is being stretched. So you're stretching your quads. And so what happens is that this sensory will come in, and it will cause excitatory, like an action potential to be generated. And you'll have one motor neuron going to your quads and then another going to your hamstrings. But we don't really need the hamstrings to contract. We need the quads to contract. So this is like, oh no, we're feeling a stretch. We need to contract so we don't, injure ourselves. So it's going to send an excitatory response to the motor neuron of the quads, causing this to contract when that's feeling stretched. But we don't need the hamstrings to do anything, so it's going to relax, for example, because those work together, obviously. And anyway, so we'll send an inhibitory signal to the hamstrings, allowing those to relax, because again, the attention is focused on the quads. So basically, it'll tell it to contract so that we don't injure ourselves by stretching too much. However, with the Golgi tendon reflex, this is basically when the body is afraid of the tendon breaking or the tendon snapping. So if we feel the stretch, if we're bending our leg and we feel a big stretch right here, what'll happen is that it'll have a sensory going up, and then, sorry, it's not drawn in here, the inhibitory, but basically what happens is it'll inhibit the muscle so it'll inhibit the muscle. In this case, it would be the quads, so it'll inhibit that and tell it to relax. So whatever the main muscle is, the main driver muscle here in this case would be the quads. So if we're stretching that a lot and the tendon is under a lot of force, then the Golgi tendon reflex will tell it to relax, so that the tendon does not break, if that makes sense. And it'll activate the hamstrings, and causing those to contract, which is, again, the opposite of the quads relaxing. And again, the whole purpose is when that tendon is under a lot of stress. So this is how we can, for example, if we're stretching our hamstring, if someone is stretching our hamstrings and they're pushing really hard, this can be activated. So if we're stretching really hard, the tendon's under a lot of pressure, it'll tell it to relax. And then maybe we relax for a second, and then we're able to go further because that muscle is relaxed. We're able to stretch it more. Anyway, so that is those reflexes. Let me make sure I got everything in there. Muscle sensory receptors, muscle spindles, and Golgi tendon organs. Okay, I guess another structure that I missed over was these intrafusal fibers, which are just the muscle fibers. There's some on the inside of the muscle spindle apparatus and some these extrafusal fibers on the outside of the muscle, or outside of the muscle spindle apparatus, but inside of this muscle. Here's another point. More muscle control means that we're going to have more spindle apparatuses in the muscle because we need more control. And yeah, but I believe that is it.